Hello everybody, my name is Malik and thank you for joining me. Today in this video, I'm going to present the research I did on the effects of vitamin C and B3 on the apoptotic rate of metastatic breast cancer cells treated with etoposide by green fluorescent assay. First, I'd like to give you a little bit of information about myself. So I was born in Turkey but raised in France, and I decided to come to America for college and to pursue my basketball career and my education. I first got an associate degree in math science in Maryland, and then I came to Tampa for my bachelor's in biochemistry, which is ASBMB accredited. For this research, my overall goal was to test the effects of vitamin C ascorbic acid and vitamin B3 niacin on the apoptotic rate of MDA-MB231 cells treated with etoposide. So apoptosis is cell death, and those MDA-MB231 cells are metastatic breast cancer cells. This research was offered through the undergraduate program of UT, and I was under the mentoring of Dr. Witherow, and I received key insights from Dr. Carastro. Some classes that helped me shape these projects were tissue culture, molecular basis of cancer, advanced biochemistry, and biostatistics. Cancer is a group of disease characterized by uncontrollable cell growth and division. And when they are metastatic, they have the capabilities to migrate into other parts of the body and develop there. There are more than 200 types. And however, there is common patterns to all those cancers. They are defined as hallmarks. So we can see them here in the red boxes. And the two white boxes refer to enabling characteristics. So most of the chemotherapeutic agents available on the market today market one or more of these hallmarks. So in recent years, combination therapy have been promoted, which consists of supplementing people that are under chemotherapeutic therapy with micronutrients. In the molecular basis of cancer class I took, we studied the effects of green tea extract on gene expression. My gene was UBE3C, which is a ubiquitin ligase. In this research, I used etoposide to treat the cells, and it induces cell death. The way it does that is by interacting with topoisomerase. Topoisomerase is a DNA repair enzyme, but it also induces DNA breakage. And the way etoposide induces double-stranded breaks is by blocking the topoisomerase 2 in a state that is not conform, and therefore it produces irreversible double-stranded breaks in DNA. So therefore it has pro-apoptotic effects. Vitamin B3 and C are antioxidants and potential adjuvants that have been studied recently. So vitamin B3 is a precursor to NAD, nicotinamide adenine dinucleotide, the most common electron transport carrier in our bodies. And it's been commonly coupled with oxygen and carbon dioxide to enhance oxygenation and increase blood flow of hypoxic cancers. That therapy is called ARCON. And vitamin C has been effective as an adjuvant for different cancers such as glioblastomas or colon cancer. So our hypothesis was that vitamin C and vitamin B3 addition will increase the apoptotic rate of cancer cells when treated with etoposide. So to articulate this research, I designed a protocol which consisted of plating cells in a 96 well plate and letting them grow for 24 hours. Then changing the media with different conditions and letting them grow for 24 hours. Then I will proceed to a apoptotic assay using a cell death marker. And that incubation period will be of 30 minutes before I subjected the plates to a plate reader analysis to quantify fluorescence. So MD-MB231 cells are metastatic breast cancer cells, and they're triple negative and highly aggressive, which makes them difficult to treat. That's why I used such a strong chemotherapeutic agent as etoposide. The cultivation condition was standard, as I used DMEM, dual Beco modified eagle medium, in a 96 well plate. The specific concentration of each component are as follow. So etoposide was 100 micromolar, vitamin C was 100 microgram per milliliter, and vitamin B3 was a 10 microgram per milliliter. To proceed to my apoptosis assay, I use the Cell-Event Caspase-3 7 Green Detection Reagent, and I dissolved it in PBS with 5% FBS to a concentration of 4 micromolar. So caspase-3 are early apoptotic markers of cells. They will fold out of the cells. They are extracellular protein that will indicate early apoptosis. So there's a peptide that was designed to bind to this caspase-3 with a fluorophore and quencher attached. But once the caspase break down the peptide, the fluorophore is released. Therefore, we can quantify apoptosis by quantifying relative fluorescence. So the statistical analysis after we quantified the fluorescence was done in RStudio. So I decided to present my results in a box plot. So the lower end represents the minimum value, the upper end represents the maximum value, and we have the quartile 3 at the upper limit of the box and quartile 1 at the lower box end of the box. The black line represents the median, and if outliers are present, they're represented by a circle. So these were our 5 main experimental groups, and ETO is our positive control. ETO VB was etoposide plus vitamin B3. ETO VC was etoposide plus vitamin C, and VB and VC were just simply vitamin B and vitamin C added to complete media. So our baseline basically represented our negative control, which was complete media only, and we normalized all the results to the baseline. Therefore, we can imagine that the value is 1 for a regular complete media and non-modified complete media. So our positive control showed a 5-fold increase in the relative apoptosis. And looking at this box plot, we can see that the medians of ETO-VB and ETO-VC are quite similar to our positive control ETO only, but their spread is much different, with ETO-VC even having 2 outliers. Interestingly, the simple addition of vitamin B3 and vitamin C seemed to increase the relative apoptosis by a factor of 2-fold. That was not something we expected when we first designed the experiment, and we are confident that there is some patterns to be explained there. Significant difference were not observed between our positive control and ETO-VB and ETO-VC, our main experimental groups. That is not inconclusive though, because as we see, there was a huge difference in their spread and values, and it's even more obvious when we look at the means of each group. So one might tend to think that there was an upregulation in relative apoptosis, but looking at the median, it's not quite obvious, and especially the standard deviation being so high for the 2 experimental groups. So through my research, I was able to conclude that vitamin B3 and C alone showed promises in upregulating apoptosis in this metastatic breast cancer cells. However, when treated with etoposide, we were not successful in identifying if there was any upregulation of apoptosis, which was our main hypothesis. So the underlying mechanisms are not yet understood of why that is happening, but some ideas have been expressed and theories have been described. So the expression of a multitude of genes characterized by cyclic AMP-dependent pathways seem to be regulated by vitamin C. And also, the activity of histones and DNA demethylating enzymes can be possibly regulated by vitamin B3 and vitamin C. So thank you for watching my video, and if you have any questions, you can email me and I'll be happy to answer you. Here are the works cited if you want to go deep into this.